him. The title of the talk is Anomalous Diffusion, and this topic, I believe it's especially valid for biological applications, although it was not very often covered on this conference. So I want to explain why, in our opinion, this anomalous diffusion is very important to biology, and this part is encoded that anomalous diffusion is everywhere, but the second part is the question where this anomalous diffusion is. And by this question, I mean not that we are not observing the anomalous diffusion, but the question to find the processes related to biology which couldn't be explained other means other than anomalous diffusion and anomalous processes. So in my talk, I will, and this is the picture how we look on our planet. So this is the Earth in the middle, and this is the model of a cell. So the nucleus is our planet, and the cytoskeleton, which is here, is the universe, and our Earth is living on this universe of cytoskeleton. This is from our recent paper, which will appear shortly in Biophysical Journal. So what is anomalous diffusion, and why anomalous diffusion is different from normal diffusion? So normal diffusion is depicted here on this left side, and it's composed in many short steps, which a particle or cell or something inside cell is performing randomly as time goes by, and these many short steps are uncorrelated, and the particle can jump forward, backward, and this results in this very irregular pattern of diffusion. Whereas if you observe that the particle which is diffusing in time stays for a long time in one place and obviously doesn't move, doesn't do anything, and you see this times often, then this long waiting periods is an indication of anomalous diffusion, and this indication of subdiffusion, the process which is sub to this normal diffusion. On the other hand, if you observe that the trajectory of your object is changing rapidly in short amount of time, and there is a long jumps in the trajectories back and forth, so this is the indication that something else going on in your system, and these long jumps indicate that there is some super process opposite to this sub process, which also indicates that there is some anomalous process, and we call this super diffusion. But sub and super diffusion, these are not only these waiting periods and long jumps. There should be something which differs from the normal process, and this something is, there is no typical scale. For subdiffusion, there is no typical time scale of these jumps. And for superdiffusion, there is no typical space scale. So this means that if you calculate the waiting periods for subdiffusion, calculate the histogram of these periods, there will be no average waiting time. If you calculate the length of jumps for superdiffusion, and you calculate the average of these jumps, this average will be infinite, and this will mean there is no typical scale, no average jump for superdiffusion. All this anomalous process is encoded in an indicator which is called the mean square displacement. So how fast the cloud of your particles spread in time. So if you calculate the squared distance from the initial position, for normal diffusion, this mean square displacement grows linearly in time. So this exponent is equal to 1. Whereas for subdiffusion, this is sublinear linear behavior, and for superdiffusion, this is superlinear behavior. So now, what causes anomalous diffusion? So actually, there are lots of mechanisms which can lead. And there is a nice quote from Lev Tolstoy in the beginning of the Anna Karenina. He wrote that all happy families are all like, and every unhappy family is unhappy in its own way. So for us, a happy family is diffusion processes, normal diffusion, and unhappy family is anomalous processes. So the anomalous process can be caused by many mechanisms, and what is worse, these mechanisms can be superimposed. They can all play on the same time. So these causes can, so subdiffusion can be caused by obstacles, by fractal geometry, by diffusion barriers, some transient binding to some bonds, chemical bonds, and also by viscoelastic response of cytoskeleton, for example. And superdiffusion can be caused by turbulence and some external driving. So here I just find some nice pictures. This is a Lorentz model of diffusion, and the particle trajectory shown here, it's bouncing in a two-dimensional space, and this is the blue dots. These are obstacles. And you see that these obstacles are randomly placed in two-dimensional space, and the particle can stick, it's bounce between obstacles, and it has hard times to escape from these cages which are formed by obstacles. But once it's escaped, it will find again place where to stuck and so on. So this causes the hindrance of diffusion, and the diffusion here will be anomalously slow. Another example which causes subdiffusion is a fractal, and this is a typical fractal. This is a tree. So there is a main branch and there is a offsprings, and if you find the diffusion over here, it will take a lot of time to diffuse to the main branch or vice versa. So this scale-free object will generate anomalously slow diffusion. As an example of superdiffusion, this is a famous example of Solomon. So you see here the turbulence in two-dimensional circulating fluid. So this is the top view, and so here the water is circulating clockwise, and this is the tracer particle. This is a particle which is passive, so it is dragged by the fluid, and the fluid is rotating in this two-dimensional space, and it forms the vortices, and the particle is trapped in these vortices and makes a lot of rotations until it escapes and gets into another trap. So depending on the speed how the water is rotating, the diffusion becomes faster and faster, but it's here it's stuck in this vortices, so it's not ballistic diffusion, this subballistic diffusion, this superdiffusion.
Question?
Yes?
Why would you call that last case a case of diffusion anyway? Because it's more like convection.
Yeah, it's convection, but it's a random. So convection is something which is dragged by the constant flow.
Yeah.
And here, this vortices they are random. So and the time which is particle spent in each rotating event is also random. So it's
still there's an external force that's driving most of the motion, right?
Yeah.
Anyway.
Yeah, but the difference is that it's more random than the usual drag. and yeah, and that's why it's not the ballistic, it's subballistic. So why anomalous diffusion is important in biological applications? There are actually a lot of examples of anomalous diffusion inside cell. For subdiffusion, subdiffusion was observed in two-dimensional plasma membranes, which is depicted here. This is the cartoon of organization of the membrane, which consists of cytoskeleton, which is this branches, and the proteins which are trying to find the way across the membrane. They can be stuck to this cytoskeleton filaments, or they can find itself in the cages, as in the previous example of Lorentz gas. So this diffusion is very complicated. It may consist of subdiffusion due to barriers. It's also known that the stickiness to this protein causes anomalous binding, and the particle needs to unbind first to proceed. There is also diffusion in the nucleus and in a cytoplasm, whereas superdiffusion in a cell caused by active transport. So there are, obviously, subdiffusion is not very good for a cell, meaning that cell needs to transport things efficiently inside to function, and subdiffusion just makes the processes slow. So for some particle to cross cell, it will need days to complete this. And the nature finds the way to overcome this. It invents the active transport. So there are a lot of molecular motors, for example, like myosins, kinesins, dyneins. And these molecular motors are designed to move along the filaments of cytoskeleton. And this is a cartoon of a cell with a cartoon of the cytoskeleton. So these long green lines are microtubules. They are long, originating from this close to the nucleus and span along all the cell. There is also a short filaments, which is actin, and it's mostly close to the boundary of a cell, to membrane. And the molecular motors, so these microtubules are polar, so they have intrinsic polarity. And some motors move from like dynein, they move to the minus end, so they move towards the nucleus, while kinesins, they move in opposite directions, and the myosins, they move in both ways. So they can move to the plus end and to the minus end. And there are also different kinds of myosins. So movement of these molecular motors, they take the vesicles of the chemicals and they move it along these lines. So this is like a highway in a city. So these are the very fast highways. This is like a local streets. So these are some experimental results from paper of 2004, and these people, they try to understand how the subdiffusion is caused by the actin network. So they extract the actin network in some substrate, and they put a particle which is passive non-reactive particle. And this particle can have different sizes. So the A is diameter of this particle. And then the particle is trying to, this magnetic bead, and using some external forces, they're trying to drag this bead along this actin mesh and see how this particle moves. So what they find in the experiment that sometimes the particle is not moving at all, and there's a time, and there's a position of the particle, and sometimes it gets stuck in one places and eventually escape from this cage, go to another place. So the trajectory of this bead is look like this. So then they extract the length of this stays of particle of trapping times and plot the histogram as a function of trapping times. And they find that this density, the probability density is more like a power law. So they feed this power law with this function, and they plot the exponent gamma here as a function of ratio of diameter of the bead, and xi is a typical mesh size of the network. So what they find is that this gamma depends on this ratio like this. So it decreases until approximately 1 and then stay almost 0. So when the mesh size is big enough, or the diameter of the bead is small, then there is no problem to diffuse in this system. So the small bead can go around this mesh easily, and that's why the diffusion here is normal. So the exponent gamma is equal to 1. When the bead gets bigger or the mesh size smaller, the diffusion is getting slower until the bead is completely stuck, so it can't move anymore in this mesh. So this is the intuitive and very nice example of the subdiffusion in an actin mesh, and what it will be for a particle to diffuse in this complex system. There's another good experiment done by Caspi and people in 2000. They put a similar bead inside the cytoskeleton of a living cell, but in this case, this bead can be taken by motors and attached to a cytoskeleton, and it can be dragged along the cytoskeleton. So they measure the mean square displacement of this bead, and they find that it is the exponent is greater than 1. So this bead is moving subballistically in this system. And why they know that this is caused by the molecular motors and the diffusion along the cytoskeleton, because when they apply the treatment to the cytoskeleton, when they disrupt the cytoskeleton, the diffusion gets slower than normal. It got subdiffusion. So this was an indication that in this system, the active transport along the cytoskeleton causes superdiffusion. Not only the anomalous diffusion is not only important inside cell, it's also important to a big object like animals. For example, it's albatross. Supposedly, it's making super diffusion flights when it's searching for food in the open sea. So it's searching in some place where it's close to his nest. Then it makes a long flight to the open sea. Then it's returned to its home. And if you measure the distribution of this length, when it touches the water, it's somehow it's reasonable to the power law, and people argue that it can be Levy flight. And the argument why they think this could be a Levy flight, it's because the Levy flight could optimize the time which needed to span to explore this big space efficiently to find the food. After this, people start looking at other animals like bees, sharks, monkeys, and even the cell as an object. It moves on a substrate with certain velocity, and then it's changed its mind and then start moving in another direction. So all kinds of big animals doing anomalous diffusion too. And even me, I'm also doing anomalous diffusion. Since this is my own Levy flight, when I finished my PhD in Germany, then I moved to Israel, and from Israel I moved to New York to do postdoc with George Zaslavsky. Then I moved from Zaslavsky, I moved back to Israel to work with Eli Barkai and Yossi Klafter. From Israel, I make another big flight to California to work with Ajay and Casey, and this is not complete. Now I moved back to Manchester, and so this is the big flights here are indications that I'm doing anomalous diffusion too. So for Levy walk, just a quick reminder that this can be viewed as a movement with a constant velocity for random periods of time, and these times have no scale. So this can be distributed by power law with exponent, and this exponent will determine which type of motion you have. So if this is between 0 and 1, this is ballistic diffusion. If it's between 1 and 2, this is super diffusion. And if it's greater than 2, you're back to a normal diffusion. But similarly, simple idea behind the Levy walk turns out that it's very difficult to write down the single governing equation for Levy walk. Only recently, Fedotov, when he was here, he submitted the paper which derives the single governing equation. So for those who want to take a look, it's here. As anomalous diffusion can be caused by different mechanisms, there are also different models. And every people who work in this area, they have their favorite models. And for us, it's continuous time random walk model. It's originated in around 1975 by these people. Oh, sorry. Montrol, Weiss, and they derived the governing equation. So for the probability function in this in Fourier Laplace space, and this probability density function, it's governed by the jump length distribution and the waiting time distribution. So the only thing which you have to know what determines the density of a spread of your particles is these two functions. So depending on the form of these PDEs, you can have the normal transport if you have this finite time length and finite jump scale. This time scale and jump scale, they will obviously determine the diffusion constant. But when your waiting time is scale free, so if, for example, it's a power law with exponent between 0 and 1, so if there is no typical time scale, but still you can have the typical jump length, you end up in this simple form of the probability density function, which when you convert to a real space, it will you will end up with so-called time fractional diffusion equation, which will involve some Riemann-Liouville operator, which is defined here, but actually this is just the Laplace transform of this. So if you compare with a normal diffusion, the only difference is this one. So when alpha is equal to 1, this disappears and you are back to the normal diffusion. And obviously the mean square displacement grow linearly with time. Whereas for subdiffusion is this sublinear linear function. Okay, so this just very simple case. And this is how the propagator looks for anomalous diffusion. So opposite to the normal diffusion, you see that there is a bigger probability to find your particle close to the where you start from. And this memory remains along the time. So it's the slowly decaying memory of initial conditions is a sign of anomalously slow diffusion. We can go beyond the monofractal kinetics. What if you have a not single particle which is characterized by a single exponent, but you have a bunch of particles and each is of different size? So as we know from an experiment of actin mesh, the size of a particle determines the exponent of subdiffusion. So if you have distribution of particles and each particle is of different size, so obviously you will have a distribution of exponents, and you can write the time fractional diffusion equation with distribution of the exponents corresponding to different size of particles. So instead of time fractional derivative with one exponent, you'll have also the probability density of this exponent, and this is a convolution between both. So if you consider the probability to consist of only two types of particles with two exponents, you'll find that your system will alternate between behavior with one exponent and the second exponent. So you can have subdiffusion which starts slower, but then it's transition to a fast diffusion. So in this case, you will have some acceleration of subdiffusion. And this is the reference for which was just accepted in Physical Review E. But what if you're exploring the space which is not homogeneous? So if you have space which, for example, your mesh of actin has different mesh size in different regions. So you will have different exponents in different space regions, and therefore you have to write down two equations. If you assume that for simplicity, for x less than 0 you have one exponent, and x greater than 0 you have another exponent. So if you solve the system of these coupled time fractional equations, starting from particles on the boundary, you'll find that as time evolves, all particles will appear in one side of a system. So also if you start from symmetric or any boundary initial conditions, all particles will end up in a part of a system which has the exponent, the minimum of two exponents. This was quite surprising because it will mean that you can separate particles in this system. So starting from symmetric initial conditions, we'll have some mechanism to separate slow and fast particles. And in this system, you can speculate that if this is the cell which is moving on a substrate of some mesh, for example, collagen, and the properties of this collagen is such that the cell is moving subdiffusively, then the stiff and soft part of a collagen will trap the cells with a different speeds.
How do you handle the jump?
The jump, well, this because of the boundary conditions.
No, at the junction between the two different regions.
What if you have a jump?
You do have a jump, right?
Yes, yeah.
How do you match them?
It's so you have to. There are two boundary conditions here. One is a flux is constant along the, and second, the ratio between values of probabilities on both sides are equal to the ratio of diffusion constants on both sides. Yeah, this was the challenge in this problem to write down properly the boundary conditions, and that's why it appears in this paper in a good journal. So before, people was assuming that the probability are equal on junction from left and right. For example, for normal diffusion, you write equality for. We show here that this is not the case for anomalous diffusion. Okay, so this was for heterogeneous environment, but when you have two different exponents in different space, but even if you have one equation like this, but the exponent is depends on x in quite simple way. So we have exponent which is constant almost everywhere except some small part. So we have some deviations in one tiny place of a space. In this case, the evolution of your particles, they all will be attracted to this point and stay forever. So subdiffusion will attract all particles and trap here. So this is not very physical. So somehow the nature should avoid this. For example, one of the mechanisms how this anomalous trapping can be avoided is by including the density effects. So when you assume that when the density of particles is getting bigger, another mechanism is taking place, and which prevents the accumulation of particles. And we ask, can the anomalous aggregation be prepared, or can we get anomalous aggregation without assuming anomalous process in advance? And the answer is yes, we can. If we assume that your particle is performing the random walk, and it has some escape rate which depends on the density, and it jumps with an exponent which depends on the age of particle. The age is so if the particle stays in some place, so it's when it arrives to some place, you assume that particle start a new life. So it's the age of particle is 0, when the particle escapes from the point. Assuming that the exponent is inversely proportional to the age of a particle is equivalent to say that the waiting time in some point is a power law with some exponent. And if this exponent is less than 1, you have, again, the subdiffusion, but let us consider the exponent greater than 1. So we have normal diffusion on normal process in advance. So once you assume the jumping probabilities as usual, which depends on the on some concentration of chemicals, then you end up in a governing equation for the density with a total escape rate, which depends on the density and on the escape rate. So for Markovian case, this is known to be just the product of these two. For subdiffusion, this convolution with a fractional operator. In general, this is not known. So if we start from mu, which is greater than 1, and we assume that the constant is the concentration is linear. Oh, sorry, this wrong. So for Markovian case, you starting from some distribution, you end up in stationary regime, assuming some reflecting boundary conditions. But when you switch the nonlinearity, you assume that A is equal to 2, for example, here. You find that all particles are again collapse to one point. So in this example, the anomalous aggregation is not imposed by anomalous process in advance. It is self-organized by the combination of the density and the anomalous exponent. So the criteria for the anomalous aggregation in this case is the ratio between exponent and the density at the critical point. So usually people observe that anomalous diffusion is transient, and there are many reasons for this. For example, the space can be just confined, so you have not enough space to explore. And then another reason is that the trapping can be destroyed by the thermal fluctuations. But other reasons, and we say yes, there are other reasons, and these reasons can be related to the nature of the random itself, random walk. So we we start from the subdiffusion model, which is and the force is acting on the continuous time random walk. So you again have this equation for the evolution of the probability density, but the probability to jump left to right is biased by the force. So you can write the total escape from point x at time t, and you end up in the time fractional Fokker-Planck equation, which has a force here, and the diffusion coefficient is determined by the typical jump length and the time scale. But in this picture, the force is acting only here. So it acts only at the moment where the particle escapes from. So this is not always fulfilled. So for example, if you think that your particle is bound to inside the cell to some chemical bond, and there is a flow which is there are a lot of flows inside cell, and the flow is dragging your trapped particle. So this flow is acting all times, not only the times as escape. So this picture will be not correct. So we need to in this picture to consider the the force which is acting all time. And for this, we assume that the escape of a particle from to the left and to the right is governed by two processes, by the escape of random walk itself and by force. And the force just modifies the escape. It makes it bigger if the force is greater than 0 to jump to the right, and make it less if the force is less than 0 to jump to the left. So in this picture, the equation, governing equation for the density looks like this, but the, the total escape is now the convolution of some kernel and the exponential which depends on the force and the density of a particle. And this kernel is determined by the waiting time of particles, and we distinguish here the waiting time of particles who moves to the right and to the left, and the survival function. So if you pass to the continuum limit, you end up with a fractional diffusion equation, which looks completely different from previous one. So we have the usual forces entering in different places. It enters here, but also enters in the exponential factors here. And compared to the previous Fokker-Planck equation for the force acting only on escape. So for constant force, what does it mean? So you end up with an equation which looks like a normal transport, but actually, is it normal at all? If you calculate the diffusion constant, you'll see that the diffusion depends on the force and on the exponent, on the anomalous exponent. So in this case, even though you observe the normal transport, normal diffusion with some dragging, your diffusion constant depends on anomalous exponent, on anomalous properties. So anomalous process is hidden behind the normal in this case. So that's why I was asking in the beginning, where is the anomalous diffusion? So anomalous diffusion can be actually in a normal transport, but you have to explore whether the diffusion coefficient or diffusion properties are still normal and not depend on some properties of which are not expected from a normal transport.
So an experimentalist, though, would be...
Yes, that's why we argue that the experiments which are thought to be normal, the experiment should, if they suspect that some anomalous process could going on there, they should carefully check the experimental results for this for these effects.
How would you do that?
Yeah, that's a good question. For example, if you have this is a constant diffusion constant, but you can try different boundary conditions in your system, trying to change the boundary conditions. And if you consider some reflecting boundary conditions in a quadratic potential, so you'll see that for normal diffusion, you'll end up with a Boltzmann distribution. In this case, you'll end up with some distribution which is not Boltzmann at all. So you'll have some peak which is characteristic to the subdiffusion, and the tails of the distribution will be in between Gaussian and exponential. So that's one possible way how you would check that in this system you don't have the normal diffusion.
How would you change the boundary conditions experimentally?
yeah, in vivo it's quite difficult, of course, but I think in vitro you can change the force profile. So if you apply not a constant force, but if you apply some laser which is creating the potential more like quadratic. Okay, so the same picture can be applied to a Levy walk, not to the subdiffusion, but to superdiffusion. And this was the study of muscles which are moving with constant speeds and on a two-dimensional substrate. And experimentally, it was found that they like to group in some piles. And when the number of muscles is getting large in this pile, they say, oh, we're too much here, so I want to move to another pile. And people measure the distribution of step lengths of these muscles, and they fit with different distributions, and it turns out that the Levy walk or truncated Levy walk is best fitted to these distributions. So we say, okay, if the muscle is doing Levy walk, what they do when they how they change their behavior with the density. And the same people, they do experiment with the muscles in a box, changing the muscle density. And what they find, if the single muscle is doing more like Levy walk with the long jumps, when you increase the number of muscles, they become to move more like a diffusion particles. So it was obvious that the behavior of the muscles changes with the change in the density of muscles in the box. And they change from Levy walk to a normal diffusion. So we say, yeah, we know such process which change from a subdiffusion to normal diffusion. So what if we apply here the same principle? And we did for non-linear Levy walk, we assume that the escape of the muscle from some point is governed by the anomalous escape, which depends on the time which the muscle spend in this point, and also with it depends on the density of the muscles in this point. And by applying the same technique, we find that by changing the density of muscles, the run length or jump length density, it make a transition from a power law to a diffusion-like exponential distributions. So the same mechanism applied to Levy walk, and the behavior of muscles in a box can be explained by this picture of Levy walk with escape rate, which depends on the density. Now I want to switch to more modeling part of what I'm doing, and this joint work with RJ and Casey, which I started in Stanford. We try to model the intracellular transport, and starting from some very simple model. And the picture which we have is a cytoskeleton network, which obviously is required for organization, structure, and transport inside a cell. And the cytoskeleton is very complicated. So you have microtubules in the middle of a cell. You have actin close to the nucleus and actin close to the membrane. So how to model this? How to approach to? And we take the same cartoon which I showed before. So this different filaments, different microtubules and actin. So they all have different lengths, different properties. We want to ask how the structure of the network influences the transport. So are there some optimal regimes for network density, distribution of filament lengths, and orientation, which allow transport to be more efficient inside cell? We start from very simple picture. This is a two-dimensional cell. This is a nucleus. And we start modeling the cytoskeleton with only one type of filaments. We assume that these are just straight lines of certain lengths. And we have a diffusion process which takes place in the bulk, in between these filaments. When the particle approaches the filament, it can bind with certain probability to a filament, and then it starts diffusion along the filament with another diffusion coefficient. So this is a composite diffusion process which also takes into account the structure of the network. And here I have to show at least one movie, because it's nice to have a movie. And so this is the picture how the particle moves inside this system. So you see it diffuses in between, then it binds to a filament, then unbinds, find another filament, go to another place. So what you can learn from this movie and simple model? First of all, you see that there are places in network where the particle wants to stay more time. It's the intersections of the filament, when there is a greater probability to attach to a network. And this is the boundary of a cell, which you don't have much space to move. And when the particle unbinds from a network, it wants to attach to, it has, for example, here it has lots of place to attach to a network. So you have to be very careful and pay a lot of attention to the intersections of filament. So we start to look at the transport with a quantity which is called reactivity. So this is a time which particle need to explore the system without the network. This is the ratio of this time divided by the time which is the same time but with the network. So if there is no network reactivity equal to 1, if the movement along the network is advantage, then this time is less, so this reactivity is greater than 1. If it's not advantage, then the reactivity is less than 1. So changing the number of filaments in our model, we can calculate that, and this is the time which particle spends on the network. So we can somehow find that indications that there is some optimal time which particle wants to stay on the network. And this is, so this is the suppose optimal time. And this is the trajectory for this optimal time. So this can be understood by comparing this trajectory for another non-optimal time spent on the network. So if you look at the trajectory in this region, you will see that the particle stays too long on the network. It's unbound from a network very rarely, and that's why it can be dragged to some unwanted part of a cell by the network without falling off. So there must be some optimal time spent on the network. But this was the diffusion on the network. But we know that molecular motors are processive, so they move with certain velocity quite long time before falling from the network. So they have some speed, which is typically 1 micron per second. And not to explore all parameter space, we also take some typical on and off parameters, so they might not be optimal as I just showed you, but these are experimentally observed on and off rates of attachment and detachment to a network. And we also assume that in the bulk, the vesicle is moving with some typical diffusion coefficient, which is also we take from measurements. Now we want to explore what the orientation and the polarity of the filament play a role in a system. So we change the number of filaments, length of the filaments, and we see that there are a lot of variability in the system. So the mean first passage time can be very different from one another configuration of a system. And the bigger the filament length, the more the variability of the mean first passage time. What causes this variability is these trapping times of the network. So if the particle falls from a filament, it spends a lot of time here. It's indicated by this residence time. So the traps are formed by the network, and the particle it has a long time to escape from this. And the fewer filaments, the longer trapping times you have in your system. Obviously, the shorter filaments, the less trapping you have. So we can play with reversing polarity of a filament, and the simulation shows that if you reverse just 25% of filaments, your mean first passing time can double. But if you, we can also explore how the multiple motors affect the transport. So by this simulation, we find that actually having multiple motors can be even disadvantages, because you rarely fall from your network. And also because you have more variability on mean first passage time in this case. And actually this was experimentally, people find that having one motors can be better than having many motors in some cases.
You mean one type of motor?
No, one motor per cargo. Yeah, of course, there are a lot of motors, and different types of motors can play. And what I call emergent super diffusion is a system we observe that the network, so we have no power loss, no anomalous diffusion assumed before in this system. So we have just normal diffusion in the bulk and velocity along the network. And the network architecture itself produces the super diffusion in this system with exponent about 144. So that's why I call it emergent super diffusion. So without the network, you have purely normal transport, pure normal diffusion. So, and with this, I want to conclude that the anomalous transport is widespread and important, and what looks like a normal transport could still be anomalous, and you should be careful with interpreting your experiments Non-Markovian and non-linear random walks allow to look at aggregation from new perspective, and simple continuum model could provide important insight into transport. And we're still working on this model, and we need more experimental input, and we were lucky to find the experimental in our university, which are these people. Vicky Allen, she's experimental biologist, and she's expert in dunning, the molecular motor which is moving. And Tom, he's a physicist, but he's working on a lot on imaging and inside single cell. So together they publish several papers on first passage times of cargos inside cell, and now we submitted a proposal together to combine our continuum model with the experimental results to bring this more close to experiments and to see what are implications of anomalous transport for these intracellular processes. So I want to thank all my collaborators from more reason to perspective. Yeah, and thank you.
Oh, are there questions? Yes.
I have a couple of questions. Have people looked into diffusion in tissues rather than within the cell?
Probably yes, but I don't remember now exactly. I have to look.
I mean, would that be an interesting question just to?
Yeah, it's obviously it's a very interesting question because yeah, people look on cancer cells, but I think it's also in a tissue. So it's not the cancer cell doing super diffusion, but I don't remember what type of experiments they were doing in real tissue or in some artificial environment.
Just one last question. From a theoretical point of view, could you do some sort of homogenization procedure if you have a random environment like the one you have here, with graphs with embedded spheres, or some sort of random environment, and do homogenization to write down an effective diffusion equation and somehow try to reduce the exponents of your sub-diffusion, because I would assume it would be sub-diffusion in that case, directly from the random properties of your medium?
Yeah, that's a big question, how to relate properties of the medium to the exponent. Because obviously some elasticity is influenced a lot.
But I guess my question could be more specific. You have these bounds for effective properties of composite materials, right? Could they be related to bounds in diffusion powers if you do some sort of analogous thing, homogenization of your material?
Yeah, I don't know, maybe.
Don't you already have to have a microscopic model to do the homogenization? And therefore you've already sort of put the physics in.
Well, in elasticity, I guess it works a little bit that way. Yeah. But here, since you have, in principle, something simpler, which is diffusion, I don't know, I haven't seen it.
Yeah. It's worth thinking about.
Yeah.
So any other questions? I have some technical ones, but we can talk about them later. So, thanks again.